Welcome, welcome, science students. Today we're going to talk about the Beadle and Tatum experiment. This is a super important piece of history in our understanding of going from how genes actually code for proteins. Now, we've already introduced the idea of the central dogma, the idea that DNA has the codes for genes within it. Our genes undergo a process called transcription where they produce a complementary RNA molecule which exits the nucleus of the cell, which then is taken to the ribosomes where it is translated in another process, and the plans from that RNA molecule are turned into proteins, which are then either employed directly within the cell or packaged up and sent elsewhere. Now, Beadle and Tatum, all right, their experiment was actually really key to establishing this central idea of biology. All right, so that's them there on the right-hand side. Just here, we have George Wells Beadle and Edward Laurie Tatum. Okay, now they investigated enzyme function on bread mold, Neospora crassa. And what they did is they actually damaged the DNA of this mold by using X-rays to cause genetic damage. The reason why they were doing this is that they were trying to confirm an earlier hypothesis by Archibald Garrod. Enzymes, the molecules in cells and in our bodies that control our metabolic activity, that control all the chemical reactions in our bodies, they're actually coded for by our genes. All right, so we have our DNA with our genes on it, and they have the plans for our enzymes, which control all the chemistry in our bodies. Now, Beadle and Tatum, they won a Nobel Prize in 1958 for their work in establishing the role that genes have in regulating biochemical events within cells. All right, so their experiment, to put it simply, is they took spores of the mold, they exposed them to X-rays to cause genetic damage to the spores. They then crossed those with wild-type spores to create a series of mutant spores. And then what they did is they grew these on minimal media. So think petri dishes, they're growing them on a nutrient, and they're looking for mutants that grows complete when it's been supplied with amino acids to make its proteins, all right, but not when it hasn't been supplied with amino acids. And from identifying those mutants that were not able to actually grow completely on minimal media that didn't have all the amino acids that they needed to build their proteins, what they were able to establish was they're able to actually find specific mutants that were not able to actually produce amino acids. And what this meant is that these mutants actually had specific non-functional genes. This let them actually establish biological pathways for enzymes in these molds. For the wild type, we have a series of enzymes that one after another produce a series of amino acids that the cell uses for a variety of its biological functions. And by using the X-rays to cause specific genetic damage, they were able to find a series of mutants where if they had damage in gene A, they were unable to, on minimal media, produce the amino acids that they needed to build cellular products. But if you gave them a media with the second amino acid present, orthonine, they were able to continue on. All right. Likewise, class 2 mutants, they were able to produce orthonine, so from enzyme A, but because they had a damaged gene B, they were not able to continue producing enzymes further down the chain unless they were supplied with that specific amino acid. And likewise, same idea for class 3 ones. Now, what that means is that they were able to establish the hypothesis as being true that one gene in the DNA of the mold actually coded for a single enzyme. All right. And we know that this is true, but not necessarily true to that detail. Not exactly. The idea at the time is that we had the prior work of Sir Archibald Garrod, a British medical doctor who was the first to suggest genes were connected to enzymes, and Beadle and Tatum's work actually confirmed his hypothesis by using genetic and biochemical studies of the mold. And they were able to, from their studies, they were able to identify bread mold mutants that were unable to make specific amino acids, and in each one where the mutation from the X-rays had actually broken an enzyme that was needed to produce a particular amino acid, unless that mold was supplied with that amino acid on its growth media, it wasn't able to actually produce that amino acid. Now, we know from later experiments today that that hypothesis wasn't 100% true. All right. But again, this is science. We work something out, and it's true until we show that we have a more updated version of that idea, or till we find out more information. We update our truth in science. Okay. So there's been some changes to the one gene, one enzyme hypothesis. All right. We recognize now that genes don't necessarily all code for enzymes. Some code for proteins. And many of the big biomolecules in our body are proteins. All right, these can be structural proteins, or they can be metabolic proteins or enzymes. Okay. The other thing we've realized is that sometimes a gene codes for a subunit of a protein, and that's because a protein itself can actually be made up of multiple chains called polypeptides. So many people call it one gene, one polypeptide, instead of one gene, one protein these days. And of course, as we've learned, genes don't always encode for polypeptides. Some of them actually encode for functional RNA molecules. So we're finding out additional details. And what this is really just doing for us is it's showing how we were able to link this idea that we have our DNA. Along our DNA, we have sections of nucleotides that make up the plans for proteins. Okay. We're able to later establish a process called transcription, where we make a complementary copy of one side of that chain as an RNA chain, which exits the nucleus, and that goes to the ribosomes of the cell. And then we're able to translate that in another process to form our polypeptide chains. Okay, and those polypeptides can be assembled into a variety of proteins. They can either be structural or metabolic enzymes. Okay, there is a lot that is actually involved in that process. And the Beadle and Tatum experiment was one of the key pieces of evidence that let us actually establish that the central dogma of cells was true. All right, so that brings us to a close right now. I just wanted to finish on a really cool piece of art. This is art by David Goodsell. Okay, he is a science illustrator who does these really good drawings of cells and other biological structures. All right, so often we describe these pretty outlandish structures and weird molecules and stuff like that in biology, and it's quite difficult to visualize them, and we make use of diagrams to do that. But even then, the diagrams are not necessarily that great. All right, David Goodsell, he does these brilliant illustrations that are really, really straightforward to understand. You can find illustrations of his for viruses, for blood cells, for a variety of different biochemical processes. I suggest doing a Google image search of his art. Thank you for watching this video. Let's get on with the lesson. Bye-bye.